schizophrenia is one of the major debilitating disease. Most importantly, this is not a disease that is say patients with the treatment will recover in a week or two. This is a lifelong treatment. It's a management rather than a cure. That's the big key difference.
Well, Lax, thank you so much for taking the time. I've been so looking forward to this conversation.
Thanks for having me here, Brandon.
Yeah, for the audience, Lax is the founder and CEO of Reviva Pharmaceuticals. He's been pioneering a lot of really exciting work in psychiatry. So I'll actually kind of hand it over to you, if you don't mind giving me maybe just a thumbnail sketch of your career.
Sure. So, I'm a translational scientist by training. I have a PhD degree in medicinal chemistry, and then working in drug discovery and development now close to 25 years, contributed to multiple INDs developed. One drug got approved currently in the market. At Reviva, we are developing two new chemical entities currently in development. So, in a career span on the last 25 years, I'm an inventor on over close to 80 granted patents and over 120 filed patents in at various stages of in a patent prosecution. So, with my career in the drug discovery over the last 25 years, and later on, a later part of my career, I turned into an entrepreneur. Started Reviva in 2006. Since then, we made significant progress. This is a developing new drug in a psychiatric space is quite challenging. So we were able to raise quite a bit of money, over $150 million to come to this stage. Now we are very close to NDA. We completed multiple trials in schizophrenia, treated over 1,000 patients with our lead molecule discovered in-house. Its drug name is bileroxazine. I am the inventor of this molecule. So this has been a very exciting journey for me, and then also quite a bit learning curve. Of course, I'm trained as a scientist, venturing into financing business as an entrepreneur. That's really a good learning, especially raising capital has been a very humbling experience.
Yeah, of course. I sometimes sit on the other side of that as well, and I fully empathize with the kind of fundraising journey. Tell me, what is unique about this lead asset?
Sure. If you look at often people think schizophrenia is a mental illness. Maybe many people take it lightly, but the reality is schizophrenia is one of the major, debilitating disease. And most importantly, this is not a disease that is patients with the treatment will recover in a week or two. This is a lifelong treatment. It's a management rather than a cure. That's the big key difference. Second thing is schizophrenia in general, mental illness. They are not, schizophrenia in particular is not a single disease. So there are a lot of complexities involved in that. Longer these patients suffer, they are likely to acquire multiple diseases. That is either partly due to organic cause of the disease, partly because of the currently available treatment drug-induced disease. But bottom line is, this is a lifelong disease in most patients. Most importantly, some of the diseases, if you look at a later part of their life, they get maybe 50, 60 years, then remaining lifespan maybe 10 years or 15 years, depending upon the individual. But here it is different. Most patients, when they get schizophrenia, they are teenagers, they are high school kids or elementary kids. So that they have a full life of additional 50 plus years to survive. This is extremely important for the individual as a functional in a society, as well as a family members in society. In overall, having an effective treatment bring back to normal these patients to normal functioning It is extremely important, but unfortunately, currently available treatments are suboptimal in many ways. There is a room to, uh, for a next generation drug to make this patient's life better. Having said that, now how big is this? The disease burden in general. If you look at the number of patients suffering from schizophrenia alone, when I say schizophrenia alone, the schizophrenia often associated with multiple comorbidities, even in the psychiatric space itself, depression, bipolar, overlap. So schizophrenia globally, around 24 million people suffer from schizophrenia. In US alone, around 4 million people. So this is a big, major burden to the patients and society. Our drug addresses the major unmet need. It is designed to address this. It is not an accidental discovery. It is we discovered in-house based on the rational drug design. The key differentiation, if you would like me to highlight, is say often we talk about first what's the mechanism, mechanistically, what it can deliver to the patient's outcome. So mechanistically, our drug addresses the dysfunctional serotonin-dopamine signaling. That's the primary mechanism for causing schizophrenia. Most importantly, next step is to every single patient have neuroinflammation at varying degrees of neuroinflammation. What we are learning in the last 10 to 15 years, especially addressing neuroinflammation in this patient population have multifold benefit. One, it can enhance the efficacy, mitigate some of the side effects what these patients experience, whether it is an organic cause of the disease or treatment, such as endocrine disease side effect, metabolic, and then most importantly, motor side effect. So combination of better efficacy, better safety would lead to better treatment adherence. Why the treatment adherence is one of the major concerns in the treatment. If you look at the statistics with the currently available treatment, around a 30 to 45% patients discontinue in acute trials, and acute trials in the sense normally it is done in the hospitalized patients. Once they stabilize after 4 to 6 weeks, they take medication at home. That requires a couple of months to years. In a 1 year trial, what we have noticed around 70% patients discontinue. In our trial, we have seen in the hospitalized patient, 16% drop of the efficacy drug. That is lower than placebo, around 50% less than placebo, other treatment currently available. In the long term, 1 year study, we recently announced the full data set early this month on June 2nd. It's available on our website. Discontinuation rate is 35% versus other standard of care currently in the market. In similar trials showed anywhere 60 to 70%. What I mean to highlight here, in a layman perspective, if someone would like to know how do I assess this drug is safe, better than other treatment, maybe one best example is look at the discontinuation rate in the trial. That would, in a high level, give you great degree of confidence how good is the drug, because if the treatment is not efficacious or treatment is having a lot of side effect or both, patients discontinue medication. So
Hmm.
that's the benefit what we have.
And what is it about this drug that um, makes it such that only half the people discontinue?
So the benefit, as I said, one of the first thing is, let's take a look at the efficacy part of it. So why efficacy is important when you look at some of the generic indications like diabetes or the some other infection disease, for example. We have a defined goal of single disease, single endpoint. This is fairly straightforward to see. Okay, you treated this patient. What is the outcome from baseline to end of treatment? But in schizophrenia, it is not like that. Schizophrenia is a multiple mix of multiple indications. Uh, current standard of care, all the standard of care approved in the last 40 years, they used PANS, positive and negative symptom scale, as a primary endpoint. What that the primary endpoint goal is that in hospitalized patients, in 4 to 6 weeks of treatment, from acute hallucination stage to patients stabilized to, uh, their severity reduced. The in reality, out of multiple symptom domains, by and large, we call it as a positive symptom hallucination, audio visual hallucination, that reduces, and then patients stabilize, they are out of danger, but they still require medication to overcome other symptom domains such as negative symptom, cognitive impairment, mood in the sense depression, by and large anxiety, depression. If these chronic conditions are not addressed well, patients often get again the acute symptom episode. We call it relapse symptom. They end up in hospital. So what I mean to say here, the primary endpoint used for evaluating approval criteria, stabilization of patients, may not be optimal. But that does not mean that there is no instrument to evaluate the efficacy. The secondary endpoints, negative symptom, and ability for a social function, there are different scales used. If a drug has on multiple symptom on efficacy, showed efficacy on the secondary endpoints, that gives to a great degree of confidence the ability of treatment to address in a broader spectrum benefit to patients. So that is a key. That is not there for most approved antipsychotics. Out of 14 drugs got approved over the last 40 years, only 6 to 7 drugs showed beyond primary endpoint key secondary endpoint called negative symptom. The remaining one showed mixed outcome in the pivotal trials. Those drugs, not very well used. So our drug, not only primary endpoint, multiple secondary endpoints showed a very robust outcome. That is the great degree of confidence, or a clinician can make an assessment how strong is the efficacy to treat broad spectrum symptom domains associated with that. Secondly, the side effects. I said motor side effect, endocrine side effect. These side effects are comparable to placebo in our case. Motor side effect is one of the irreversible side effect when patients with some of the medication after couple of months, it's really problematic in some patients. Our drug, by treating over 1 year period, we hardly seen any motor side effect. That is a really, we believe, benefit patients. Second, metabolic and endocrine side effect. When I say endocrine side effect, most drugs currently in the market as antipsychotics, they increases or imbalance hormones. Hormones are prolactin is one hormone, thyroid hormone. Imbalance in these hormones is one of the side effects what we see, sexual side effect. Sexual side effect with some of the drugs are very serious problem. uh, these side effects lead to discontinuation. So our drug, these side effects are comparable to placebo, are a very benign. So combination of broad spectrum efficacy and then good tolerability and then benign side effects profile makes this drug, very uh, what you call attractive for physicians to prescribe this chronic conditions.
I'd love to maybe actually just kind of get a little bit of the backstory. Actually, how did you come across or discover this?
If you look at my background, back in mid 90s, we used to have a kind of an early stage. At that time, it was a widely what not widely used. I would say it was a booming space for high throughput screening, combinatorial chemistry, basically how efficiently you put the throughput in the drug discovery. I I was involved in that kind of research. I was involved in screening developing molecules for using the technology. So in the course of in multiple areas when I was working on, I realized that psychiatric space is a very big unmet need, and often people at that time, uh, most companies were focused on started to beginning to focus in mid around 2005 2006. Uh, uh, you know, Oncology is a kind of a more attractive, possibly the reimbursement related. Second one is a oncology space and a orphan designation related space. So psychiatric space is a big unmet need. And then I thought developing a drug, it is overcoming what we learned from the profile of the current treatment. If we are able to come up with even a slightly improved profile with addressing some of the unmet need related to efficacy and side effects, drug can become a blockbuster drug, or it can benefit millions of patients. So another thing is if you look at in even now the recent drug approved Cobenfy, these drugs were discovered prior to 2000, prior to genome, information available. That means when the drug developed, the optimization of these molecules were locked in prior to genome. So now we know post-genome era, we know much more about some of the key receptors. What we optimized in our case, like 5-HT2B, is highly expressed in microglia. It is one of the key receptor of target mediating neuroinflammation, and then also chronic condition such as negative symptom mood and cognition. Our drug has activity for these receptors almost 3 to 4 times more potent than dopamine activity. And then we believe that it's very well translated in the clinic, because we have seen in two large trial with 400 plus patient population, seeing robust efficacy or a treatment outcome in reduction in inflammatory markers. That is we believe related to potent activity for 5-HT2B, and then also the strong efficacy on the secondary endpoints are demonstrated in two trials or three trials, such as for negative symptom, social cognition, or a functional outcome related to a great extent differentiated profile for the 5-HT2B activity, serotonin 5-2B receptor, 5-HT7 and D4 compared to other antipsychotics in the market.
How did you decide that you wanted to actually start a biotech yourself? Like where did that motivation come from?
I was uh, I joined a company in the early my career. This company called Xenoport. I don't know whether you've come across that name. It was a Bay Area-based company. I was an early employee. I believe I was the second employee. Certainly, under 10 employees joined, and then it was a great learning experience for me. A small company from a discovery to clinical development to phase two and then beyond. I I I learned a lot in that company. See, but then I believe I contributed. I was the key contributor for success of the molecule that's currently in the market. Of course, there are other my colleagues as well worked in the same molecule, but I believe I contributed significantly for advancement of discovery as well as a scientist, bench level scientist. So, bottom line here is that focus in that company was to come up with a new molecule and a protein target in the gut, make the drug more absorbable. And also later on, we involved in designing molecules to selectively target particular organs. So then, after working for Xenoport for, I believe I worked for close to 6 years, and then joined another company called RX Therapeutics. There, my work was focused on designing molecules to overcome some of the metabolic pathways related issues. So if you look at the currently available treatment, majority drugs are cleaved by liver enzymes, especially some of the liver enzymes called 2D6 or less expressed enzyme, a lot of variability. That information was not available prior to 2000. Now we know much more about it. So there our focus was to design a drug, overcome these less preferred pathways because that are prone to cause drug-drug interaction side effect. So I learned a lot about how to design molecule to overcome metabolism-related issues. In the first company, focus on developing a drug, how to improve the absorption and selectively target brain or other part of the tissue. The next comes here. If I'm able to come up with the next generation molecule, improve the pharmacology in general, then it is a complete discovery. So that's how I Reviva started. So one thing is why schizophrenia? The next question. So schizophrenia, if a drug, if you look at, I mentioned about 14 drugs got approved currently in the market. The profile of the drug mainly focused on only dopamine serotonin target or pharmacology primary target, but their pharmacokinetics profile, absorption are very poor. And then especially when a drug is taken lifelong, those properties matter a lot because our goal is to patients have to take medication for lifelong. They need to be comfortable and then less side effect, and then it is convenient. And then we need to be mindful that as patients get old, they get multiple other indications. If drug is not compatible enough to take with other, like lifestyle disease, like diabetes medication, dyslipidemia medication, if drug is not compatible, then there are issues. So by addressing the metabolism and then absorption, it can solve 50% of the problems this available drug. At Reviva, we address the primary pharmacology efficacy related. We address the absorption as well as the metabolism. When I say metabolism, I talked about the receptor selectivity improving to 5-HT2B, 7 and D4. With respect to metabolism, uh, main problem with some of the widely used drugs currently are they are 2D6 or a less one CYP1A2. These liver enzymes are less expressed, and then patients have a suboptimal level of that lead to drug-drug interaction. We made such that our drug not going through that metabolic pathway by primarily cleared by major expressed enzyme that increases the safety significantly. And then absorption wise, our drug has absorbed almost like a 75%. So once a day dosing, and then what you call more safer drug metabolism plus improved pharmacology to target chronic condition, negative symptom cognition, and inflammation as a package, this is a better drug to treat. Maybe lifelong one can take this drug without having much problem.
Mm, that's amazing. How I guess like there's been a lot of recent progress in schizophrenia research. Can you maybe talk to me about what it's been like running these studies kind of concurrently with all the kind of activity that's been live?
So if you look at the progress in new drug development in this space, so sometime in early 2000 started, we call it as NMDA receptor target, and then a couple of companies tried in that. And then there were mixed results. Now, that is not a very attractive target, I believe. I don't see many companies, hardly any companies pursuing research in that target. Now, after that, early 2010, around that period, the glutamic receptor is another target we tried as a new target that also did not give a very robust outcome. And then up lately, muscarinic receptors have been tried, like a Corona Therapeutics, Cobenfy got approved recently. But in the similar line, there are other 2 or 3 companies pursuing a clinical stage of muscarinic receptors that did not give a expected result in the clinical trial. So again, there are various reasons for that. I may not be able to comment on our competing company's product, but what I know as a scientist, uh, at what we believe here at Reviva as an innovator in this space. So whether it is a muscarinic or a NMDA or nicotinic receptors, I think they are downstream target of the dysfunctional derived from mainstream target dysfunctional dopamine serotonin signaling. If we are able to come up with a better drug to safer drug to modulate these two signaling cascade, the downstream marker muscarinic or acetylcholine we call it, that are muscarinic, nicotinic, NMDA, GABA, they to great extent or a full extent gradually they will be also optimized in the brain. So that may be the best approach to treat, proven approach. And then often we call it as a old drug target, dopamine target, serotonin. It may not work. Many companies will promote that way. I do not believe in that because we need to carefully look at it is not the target dopamine serotonin. There are different sub-receptors are there. So what is preventing them or what is lacking in existing therapies are off targets. Often nobody talks about off target. Off targets are limiting the therapeutic balance. What's that mean? If you look at the receptor profile of a drug, it may be dopamine serotonin, but if you look at the off target, like a cardiac adrenergic target or a muscarinic antagonist that cause GI side effect or a adrenergic cause cardiac side effect. All these things limit the dose. So when you put a low dose, you may not be able to see therapeutic benefit or at a therapeutic dose, if you have a off-target activities are higher, you see a lot of side effects. That often it is not highlighted. What we believed in Reviva, what I personally believe, after working in 25 years in this space, it is not the dopamine serotonin target. It is the off target. If you can shave off target activity and then improvise some of the selectivity for the serotonin target, especially neuroinflammation, we can come up with a better drug. That's what we did at Reviva. So when I say that shave off target, if you look at our drug has hardly any appreciable activity for receptor or adrenergic that cause cardiac side effect. We haven't seen in any clinically significant any cardiac related problem in the by treating over 1,000 patients. On the other hand, if you look at some of the muscarinic agents trial, newer targets, or in the nicotinic target back in 2012-13, at the forum and Target pursued this target. There are a lot of cardiac side effects reported, increase in heartbeat or blood pressure, all these things, variability. We don't see that clinically significant in this. It is much more safer. So what I mean to say here is uh, there may be other approach to pursue muscarinic NMDA or a nicotinic, but ultimate goal is to have optimizing the serotonin and dopamine. If you can come up with the right mechanism or a pharmacology that directly modulate the serotonin dopamine level in the brain rather than indirectly modulating dopamine serotonin through muscarinic, nicotinic, or NMDA, you may be able to come up with a better drug directly modulating. That's what we did at Reviva.
Yeah, I think it's pretty amazing I guess the suite of considerations you've designed into the into the drug you're developing here. Now, one of the things that I know we've spoken about a little bit before is kind of a transition towards precision psychiatry.
Yeah.
and biomarkers. I'd love to hear you just speak a little bit about your opinion on where that's all going.
Okay. So again, in the first part, what you said, precision in medicine, in psychiatry, and biomarker. Biomarker, it is a fairly defined space, fairly defined space in the sense some of the biomarkers may not be specific to some of the symptom domains in psychiatric, but having improvement in those biomarkers certainly give a great degree of confidence to physicians as well as regulatory authorities whether drug is in the right direction or not. We may not be able to get label claim for that, but there are enough scientific evidence to, professional in this space can make a reasonable judgment while prescribing. That is certainly useful. I think we are the first company to use multiple biomarkers, not only blood-based biomarkers. They are not very specific to particular symptom domains. They have a broader effect. With respect to some of the side effects, they may be specific to a great extent. But in general, holistically, biomarker, reducing blood-based biomarker, it's a great tool to assess both broad efficacy as well as safety that we have. Besides that, we use digital biomarker that we believe has a more significance in the coming years. What's that mean? Schizophrenia patients, they have a signature pattern of the way they speak. We call vocal biomarker that a computer machine learning can detect in a millisecond precision. What we did in our trial, in the two trial, one is long-term study, open label, and then when I say open label, it has more importance in open label than the double blind. Why? Because in the double blind, we have a placebo control. When you have a biomarker, you can compare with the placebo. In the open label, even though drug may have a good efficacy, often we discount it because there is no placebo. If you have a biomarker that is independent of clinician assessment, that gives a lot of credibility for the data. What we did, we have used the biomarker, digital biomarker, to evaluate negative symptom. That's one of the difficult part to address. So we have seen what we did. It's called enrichment technique. So in real world, in the schizophrenia negative symptom, there is no such thing called pure negative symptom patients you find. Some people may argue that we find negative symptom. To my knowledge, it's not practical to find a pure negative symptom. There is always a overlapping of positive symptom to some degree and then other symptoms. What best we can do in the clinic, so use biomarker. You bring maybe on a 1 to 10 scale, maybe 8 a high severity of negative symptom. You identify using biomarker and then do the trial in that patients. What we did in our trial using that biomarker, out of 411 patients enrolled in schizophrenia study, double-blind study, we identified 229 patients, very high degree of negative symptom, and then remaining one, maybe slightly less negative symptom. So the bottom line here is this is an enrichment technology. So you randomized 411 patients, and then you did the study. Then total negative symptom pair outcome 2-point separation from placebo. That's really good informatics. Now, if you look at very high degree of negative symptom patients, what would be the outcome? If the treatment effect is very good in those patients, we should expect to see higher separation. We have seen in our study, in those 229 patients with a high degree of negative symptom, almost double the efficacy. That means drug is working for negative symptom very well. That is the biomarker we use. Now, the last part to that, when you ask, people use a precision medicine. Okay, so when people say that, okay, I have a biomarker, I can go and treat only negative symptom patients without this drug using this biomarker. It is a good and academically, but in the real world, developing a drug and then developing as a treatment may be very difficult. It's not a commercially viable. So what's that mean? So You need to have an identified, approved tool for identifying in a general setup. That is, may take years to come. Second thing is, then you will define only small patient population with a 500 million plus cost of developing. It's simply economically not viable. I don't know anybody can develop that kind of thing, or a schizophrenia. Again, in the in a innovation world, nothing is impossible. For me, that is an uphill task of considering the development cost, and then outcome economic. The cost may be couple of $100,000 per year. It may not be affordable for other patients. So there are some practical difficulties going in the precision medicine. I do not have a clarity how anyone can make a affordable treatment.
Yeah. Well, Alex, maybe in the last kind of minutes here, I'd love to hear what's next. It sounds like you're almost at the finish line here.
Sure.
What's next for you and your programs?
See, what we are doing at this time, again, we are very excited about the data, the quality, and then multiple domains, a robust efficacy, and the biomarker data available. And we have two randomized trials, successful trial, and then one long-term safety, over 1,000 patients treated. We believe we are at a stage can reach out to agency for a next step for NDA. Again, in the meanwhile, we are preparing for a second study as well, confirmatory study, phase 3. We do not know at this time. Based on the available data, we believe we met the regulatory criteria. Since we haven't pre-negotiated with the agency, now it is a time to reach out to agency, ask guidance. So we are exploring the next options, available options for taking this molecule to NDA. So in the in the near future, we will know publicly what would be the next path. But regardless of that, whether we do the require second study or the current data is sufficient for NDA, we will have an answer soon. But the data what we have is really exciting. I believe if the drug comes to market, millions of patients will be benefited.
Well, that's all incredibly exciting, and congratulations again on an inspiring journey and tremendous progress.
Thank you for the opportunity.
Yeah. Well, Alex, thanks so much for taking the time to be on our show. I've so appreciated this conversation.
Thank you. So let me know if you have any questions or